Hi, my name is Shivani Muthukumar, and my project is the identification of microRNA that may inform increased risk for progression of GERD and H. pylori to gastric cancer. Gastric cancer is a disease in which cancer cells form in the lining of the stomach. Gastroesophageal reflux disease and Helicobacter pylori are both risk factors for gastric cancer. 1 in 5 adults, or 20% of the world's population, have GERD, and 50 to 70% of the world's population have H. pylori. These large numbers of GERD and H. pylori patients potentially translates to a large number of patients progressing to gastric cancer. MicroRNA are the single-stranded, non-coding parts of genes that are generally 22 nucleotides in length. They control gene expression by binding to a messenger RNA in the cell's cytoplasm, and they play significant roles in biological processes such as proliferation and metabolism. There is also increased evidence that shows that microRNA dysregulation plays a critical role in the development of a number of diseases. So my hypothesis is that there exists a set of microRNA that is associated with GERD, H. pylori, and gastric cancer. The identification of this set of oncomicroRNAs linked with these three diseases can spur the development of strategies to neutralize them and potentially reduce the incidence of GERD and H. pylori-associated gastric cancer. For my methodology, I searched through NCBI and MIRNet to identify differentially expressed microRNAs in gastric cancer, GERD, and H. pylori. I then tried to identify an intersection set of the differentially expressed microRNA in all three diseases, and then I analyzed the significance of that intersection set in relation to the diseases. Here is my methodology a bit more in depth with the specific criteria that I had for my datasets and the specific mathematical functions that were used. For my results, I was able to find this dataset on NCBI that lists microRNA expression data in 24 gastric cancer patients and 6 non-cancer patients. With this data, I was able to make this volcano plot that you see to the right. Here, the y-axis is a negative log base 10 of the p-value, which means that points that are above this horizontal line right here have a p-value smaller than 0.05. And as you ascend the y-axis, the p-value gets smaller. So again, anything above this horizontal line is significant. The x-axis is a log base 2 of the fold change. The fold change is the ratio of the means of the two groups, which means that points that are to the far left of the plot or far right of the plot have a large fold change and either have increased expression or decreased expression. Points that follow both requirements are areas of interest and identify differentially expressed microRNAs. So these points that are blue are significant and downregulated, and these points that are red are significant and upregulated. I was also able to pull a list of all of the identified dysregulated microRNAs from this plot. For H. pylori, I was able to find this dataset also on NCBI that lists microRNA expression data in 10 H. pylori negative patients and 9 H. pylori positive patients. The formatting of this data was non-coding RNA profiling by array, which gave me access to the GEO2R tool, which performed the log functions necessary to create this volcano plot that you see to the right. This volcano plot is very similar to the one that was just shown, as the y-axis is a negative log base 10 of the p-value, and the x-axis is a log base 2 of the fold change. So again, here, any points that are blue are significant and downregulated, and these points that are red are significant and upregulated. I was also able to pull a list of all of the microRNAs that were identified as dysregulated from this plot as well. Here, Table 1 shows the common microRNAs found between the gastric cancer and H. pylori patient datasets. So from the two volcano plots that were just shown, I found this list of 11 microRNAs that were found to be differentially expressed in both datasets. Most of them were also similar in the way that they were either downregulated in both or upregulated in both. This next set of results is separate from the two volcano plots that were just shown, as I was not able to find a dataset for GERD that fit the criteria that I needed. Here, I searched the MIRNet database for gastric cancer, GERD, and H. pylori. On MIRNet, when you search for a specific disease, it returns to you a list of microRNA that is associated with that disease, along with literature of where it was gotten from or found. So once I had a list of microRNA that was associated with gastric cancer, GERD, and H. pylori, I used Excel functions to find ones that were common between any two diseases and ones that were common between all three diseases. And I displayed that on this Venn diagram that you see here. So from the MIRNet search alone, we could see that microRNA 148A, microRNA 210, and microRNA 143 are all associated with gastric cancer, GERD, and H. pylori. One thing I wanted to point out is, again, from the MIRNet search, we know that microRNA 148A is associated with all three diseases. MicroRNA 148A was also found to be differentially expressed in the gastric cancer patient dataset that I had analyzed. And microRNA 148B, which is in the same family as microRNA 148A, was found to be differentially expressed in the H. pylori patient dataset that I had analyzed. So I wanted to go a bit into the identification of microRNA 148 as a potential marker of H. pylori and GERD-associated gastric cancer. Again, microRNA 148 was found in the MIRNet search intersection set of all 3 diseases and identified as downregulated in the gastric cancer and H. pylori patient datasets. MicroRNA 148A also controls mRNA that is involved in biological processes that are associated with many cancers, such as proliferation, cell cycle progression, invasion, and metastasis. The microRNA 148 to 152 family also may serve as prognostic markers of autoimmune illnesses, chronic inflammatory disorders, which both GERD and H. pylori are, and various cancers. So in conclusion, microRNAs common to all 3 diseases were able to be found, and the identified oncomicroRNAs have the potential to be used in clinical research as biomarkers and therapeutics. Both microRNA repressors to silence the gene function and microRNA mimics to overexpress the gene function have been reported to have clinical applications in peripheral tissues. And numerous studies have demonstrated that manipulating the expression of specific microRNAs can change a drug's sensitivity, or that microRNAs themselves play a biological role in the body's resistance response. Limitations of this project include that it was limited to published publicly available data and does not account for additional oncomicroRNAs that remain to be identified or have not been published. Future research would be to continue to expand the search to include more data to identify additional oncomicroRNAs, and potentially expand the search to include mRNA data or RNA sequencing data to find the differentially expressed microRNAs. Here are my references, and I thank you very much for your time.